In the second lecture of your cells unit, I'm going to be talking about the two different types of cells that we come across, the two different categories, and then I will go into a lot more depth with one of the categories, and we'll save the other one for an in-class lecture. First off, let's talk about the cell categories. How do we tell cells apart, and where do they belong into the two major categories that makes up what cells are? You have two options. And it all depends on whether or not you have this one very important organelle. Now, first off, you can either be a prokaryote or you can be a eukaryote. Those are your only two options. You're either one of them or you're the other one. And the defining feature of these, you can do just one simple question. Do they have a nucleus? If you say no, then you are going to be a prokaryote. If you say yes, then you are a eukaryote. And that's as simple as it goes. Now I'm going to give you a few more little bits about it, but that's basically what this boils down to. You are a prokaryote if you don't have a nucleus. You are a eukaryote if you do have a nucleus. Simple as that. Just so you know, though, a little bit more about what it means to be a prokaryote, let's reiterate this, so do not have a nucleus. And one thing you could take off of this, pro means before. So what this basically means is that these cells evolved before the nucleus. So these are really old. They were around way longer than any eukaryotic cell that has a nucleus. Prokaryotic cells are mostly small and simple. Just like with everything else in life, there are exceptions to this, so they don't all fall into the mostly small and simple category, but most of them do. And then the genetic material, the DNA, is of course not contained in the nucleus. So those are your common characteristics. Now, on the flip side of that, eukaryotes, and we will spend the majority of the next lecture talking about these, eukaryotic cells have a nucleus, and eu means true. Just in case that helps you kind of remember what it is, a true nucleus is usually how I recommend you remember it by. These are usually larger and more complex cells, which makes sense because we fall into this category and we're way more complicated than a simple bacterial cell, which would be considered a prokaryote. And then the genetic material is located in the nucleus. Now, for the rest of this video, I'm going to concentrate on prokaryotic cells, and I'm going to use one of the diagrams in your unit notebooks. If you would look over onto page 15 in your unit packet, I want you to go through, and as I go through each thing, I want you to color in this diagram so you can see what it is I'm asking you to be aware of for prokaryotic cells. So use page 15, the prokaryotic cell diagram, to go along with this next part of your lecture. Prokaryotic cells, simply explained by using bacteria. Everybody's familiar with bacteria. I'm sure everybody has had food poisoning at some point in their life, and that's usually caused by a bacterium. I'm going to talk about their bacteria's cell structure versus an inside and an outside way. So I'm going to talk about everything that's inside the cell, and then I will talk about the stuff that's on the outside of the cell. For the inside of the cell, one thing that you will encounter is called a nucleoid. Now, prokaryotic cells do not have nucleuses, but to kind of continue on with that theme, they have what we call a nucleoid. Even though this is not a nucleus, DNA is generally confined to this central region. You're still going to be able to tell when you're looking at a bacterial cell where its DNA is generally going to be located. It's still going to be a dark substance in sort of the center of the cell. However, it's just not going to have a nuclear membrane. The DNA that you find here is usually circular, meaning a circle. It's a single molecule and it's a circle, circular DNA. And just to reiterate this, a nucleoid is not bound by a membrane. The DNA is free to socialize with the rest of the cell. It is not cut off like it is in a eukaryotic cell. However, you can still see a very defined place where it typically is housed. Now, a nucleoid isn't the only place that you will find DNA in a cell. You can also find DNA in something that we call a plasmid. And this is a small piece of DNA that is separate from the nucleoid. It's an extra little bit. When we get into biotechnology, I will come back to plasmids, and hopefully my plan is that you will get to work with plasmids a little bit. And you might get to do what we call a transformation with them. So keep your fingers crossed that I actually get to do that. A ribosome is also found in a prokaryotic cell, and they do exactly what they do in our cells. They synthesize proteins. Same job, no matter where they're found, they still synthesize proteins. And a prokaryotic cell also has cytoplasm. And this is the fluid of the cell. That's the structures inside. So you have a nucleoid, a plasmid, ribosomes, and cytoplasm. 4 things. I think you'll be fine with those 4 things. Now on the outside, this is where it gets a little bit more complicated, especially depending on what bacteria you are talking about. And I will get to that when I get to the part of the outside structure of a bacterial cell that's different between one type of bacteria and another type of bacteria. Now, one thing they all have, and we have them too, are cell membranes. Can't get away from those. Still got them in here. Cell membrane's job is to regulate what enters and leaves the cell. It will cover the cell completely, and it will keep the outside world out and the inside world in, unless it is allowed to come or go. It consists, just like it does in our cells, consists of a lipid bilayer, and we'll talk about that when you get into class, when we get into eukaryotic cells. I'll leave that for a class discussion. The next thing that it has that we don't have, but plant cells have, are cell walls. These maintain the overall shape of the cell, and it protects the cell from its environment. It's a little bit more rigid. It takes a lot more stress. It takes a lot more of the brute force, and so it can protect the cell a little bit better. That's why bacterial cells are sometimes so hard to get rid of, because you've got to get through all these different layers to get to them, and they have so many protection mechanisms that help protect them. So this is just one more. Not only do they have the cell membrane, they have a cell wall. Now on top of that, I'm not done yet, you have an outer membrane. This is only present in gram-negative bacteria. Bacteria can be classified as gram-positive or gram-negative, and there's certain things about those two things that we can talk about in class. But this is only found if something is termed to be a gram-negative bacteria. If I'm gram-negative, I'm going to have 4 different layers. Gram-positive only has 3 different layers. An outer membrane is just like a cell membrane. It's still a lipid bilayer that surrounds the cell. Acts just like a cell membrane, it's just on the outside of the cell wall. That's basically what it is. And you only have to remember that it's there if I ask you about a gram-negative bacterium. Now the last layer of a bacterial cell is called a capsule. Now these are the cool parts. because this is what allows bacteria to survive in almost any condition you put them in, unless they're there for an extended period of time. And this is what makes bacteria cells really tough to treat sometimes, is the capsule that they have. A capsule is a jelly-like layer that surrounds the cell. And what it does is it protects the cell from killer forces. And I'm talking about your immune cell or an antibiotic that you treat certain bacterium with. It causes a lot of things to bounce off or not to be able to attack the cell. So it's a really, really cool little coping mechanism that bacterium have. Now, along with those possible 4 layers, either 3 layers or 4 layers, depending on what type of cell, what type of bacterium you're talking about, you can also have projections that come out of the cell, and I'm going to call them appendages. They're still going to be on the outside of the cell, so that's why I'm going to talk about them here. And think of them as like your arms or your legs. They do certain jobs, but they're going to be sticking out of the cell surface. One of the appendages you can have is called a flagellum. These are whip-like projections that are used to move a cell. Cells can have 1, 2, 3, 4, or as many as 100. You can have a lot of flagellum or flagella, depending on what your job is. The one you're going to most be associated with is a sperm cell. Sperm cell has 1 flagellum on the back so that it can propel itself forward. The other projection, the other appendage that you can talk about is called a pilus. A pilus is a projection of the cell membrane through the other layers, and then it goes out to the top. What these do are they serve as attachment points. And what I mean by this is they can kind of act in 2 different ways. Some bacterial cells will use these to create a parasitic relationship with a host cell. So it'll just kind of attach itself and it will feed off of that cell. It doesn't really harm the cell per se. It'll just attach itself and it will use that cell's mechanisms for food. It will take in a lot of the environment from that cell. They can also use a pilus to transfer genetic material into a host cell. Off the top of my head, I can't think of a good example of a bacterium that does this, but a virus that does this, and viruses are not considered to be prokaryotic, but for an example, viruses do this. One big example of a virus that does this is HIV, the virus that causes AIDS eventually. What it will do is it will attach onto a cell and it will inject the cell with its genetic material, and then that genetic material will then hijack the cell and make it do what it wants instead of what it needs to do. So some bacterium have that job as well. That concludes this lecture. Bring your notes to class, and I will see you soon.